All right, everyone, we are live. A very good afternoon and warm welcome to Shri Chaitanya. Students, this is your NEET Game Changer series for 10 days and today is our 9th day. Basically, what we are doing, we are solving 25 questions per session and on daily basis. And these 25 questions are not just the 25 questions. These are basically picked up from the last 10 year of previous year questions. We have finalized the concepts from which, you're going to get question every year. And those concepts, from those particular concepts, we have prepared certain questions, okay? And these questions we are solving on daily basis. So today is the day 9 of our game changer series. Let's get started, everybody. First thing first, do hit the like button and share it with all your friends, and then let's directly jump into the questions. The schedule for the day is your classes starts from 12 p.m. to 5:30 p.m. Today, this particular class is of botany. I'm Beena Yadav. I'll be taking care of your botany part. And the class timing is 3 to 4. Tomorrow will be your last class. From 4:30 to 5:30, we have class by Pawan Kasu sir, and the class will be of chemistry. Okay. Hello, Rupa. Good afternoon. Yes, Ram Charan, class has been started. So everybody do hit the like button and share it with all your friends. And if you are new to our channel, then do not forget to subscribe to our beautiful channel. See, on 5th of May, when your exam will be over, sharp at 5:30, we will be live also to discuss the answer key of your paper, as well as the difficulty level of the questions, as well as the cutoff that we might expect, okay? So be present on 5th of May, sharp at 5:30. Okay, guys? Okay, let's get started. Your very first question on your screen. Students, first question on your screen, mitosis is characterized by mitosis. Okay, if I speak about cell cycle, we discuss two things. We can have two different type of cell division. First is mitosis, second is meiosis. So, in this question, the question is about mitosis. Mitosis is it reductional division? Is it both reductional as well as equal division? Rajeshwari, good evening. Abhisha, great answer. SD, hello. Monica, great answer. Rupa, correct answer. Rest of the kids, come on, make it fast. Is it equal division? Is it pairing of homologous chromosome? If I speak about mitosis, we also call it equational division. Equational. Why equational? Because the number of chromosomes will be equal to the parent, okay? Let's say I'm having a diploid cell. The number of chromosomes are 2n, right? Just after mitosis, it will produce 2 daughter cell, and every cell will be having presence of 2n number of chromosomes only, okay? So that is why we say it is equational division. Answer is option number B, which is equal, okay? Yes, plus for everybody. Can we have plus for in this question? Ram Charan, my dear student, reductional division is your meiosis, right? In case of meiosis, we're going to see the number of chromosomes reduces to half. If my parent cell is having 2n number of chromosome, then just after meiosis, I'm going to see n number of chromosome, all right? So from 2n to n, I'm going to see, right? Yes, so mitosis is your equational division. Clear, everybody? Moving on to the second question. Come on, kids. Rebak, hello. Arjun, ready, hello. Tilak, hello. Come on. Next question, kids. Next question on your screen. Meiosis in diploid organisms results in. Meiosis results in what? So we have two kind of division. We have number 1 mitosis, we have number 2 meiosis. Mitosis, as we discussed right now, that mitosis is equational division, wherein your number of chromosomes will be same that of parent. Okay? Yes, but in case of meiosis, the number of chromosomes will be reduced to half, just half. Correct answers, Rupa, Arjun, Monica. Yes, kids. So basically, it is used for production of the gametes. How come? Let's say this is my male parent, this is my female parent. Male is having 2n number of chromosome as 10. Let's take a random example. Your female is also having 2n is equals to 10. All right. Now, if there is meiosis happening, which is called as reductional division, number of chromosomes will be reduced to half. So it will be n is equals to 5, right? Here also we're going to see n is equals to 5. When I want to make gamete, there will be reductional division, so that when this male and female gamete is going to combine, I am going to have n plus n condition, which is 5 plus 5, again 10. So basically, I'm able to restore the ploidy of the organism just by meiosis only, right? So of course, production of gametes, it is absolutely essential. Reduction in the number of chromosome, of course, it is required. Introduction of variation. See, whenever there is a meiosis, that is linked with crossing over. And in case of crossing over, there is exchange of a segment between non-sister chromatids, okay? So basically what is happening, your new combinations will be produced. Whenever your new combinations are produced, that brings us variations. And that is very important for evolution perspective. Clear, kids? So it is your option number D, which is all of the statements are correct. All right, next question on your screen. Come on. Next one on your screen. An irreversible or permanent increase in the size, mass or volume of a cell, organ or organism is called as. The change is irreversible, all right? It is not a reversible change. It is irreversible change. We cannot reverse it back, okay? And there is permanent increase in the size, mass or volume of the cell. What do we call it? Do we call it growth? Do we call it de-differentiation, differentiation or development? It is called as, Tilak, great answer. Arjun, great answer. It is called as growth, okay? So growth, basically, my dear kids, if I speak about growth, we can define growth in the case of living as well as non-living, right? If I speak about living organisms, so the growth will be, there is basic fundamental difference between the growth of these two. In case of living, the growth is your intrinsic growth. Intrinsic, which means internal growth. Basically, the protoplasm of this cell is going to double, all right? But if I speak about non-living, the growth will be extrinsic growth. It is extrinsic or external growth. If I speak about living organism, the growth is basically irreversible. I cannot reverse it back. But in case of non-living, I can reverse it back. It is reversible. Clear, kids? Okay, so this is basically defining your growth. Clear? Yes. Moving on to the next question. Very interesting question. It is match the column type of question. We have column 1 and column 2. In column 1, we have the very first option, which is molecular oxygen. Then we have electron acceptor. Then we have pyruvate dehydrogenase. Then we have decarboxylase. Okay, molecular oxygen, alpha ketoglutric acid, nothing to do with it. Hydrogen acceptor, yes, molecular oxygen is terminal hydrogen acceptor in case of electron transport chain. All right. Now, the next option is electron acceptor. Is it cytochrome C, acetyl coenzyme A, or alpha keto? It is your cytochrome C. Pyruvate dehydrogenase will act and it will form acetyl coenzyme A. And decarboxylation will be happening into your alpha ketoglutric acid. Thereby, alpha ketoglutric acid is my 5-carbon compound. And it is the only 5-carbon compound in entire Krebs cycle. Rest of the molecules are either 6-carbon or 4-carbon compound, right? So answer is option number 1. Great. Priya, Tilak, Rupa, Ramcharan, Grace. Clear? Great, great, great. +4 everybody, +4. Perfect answers. Come on, next question. Select one which is not true for ribosome. Come on, not true, which means we need to find out the incorrect statement. Not true with respect to ribosomes. They are made up of 2 subunits. We have one small subunit, we have one large subunit, right? It forms polysomes, specifically in the case of prokaryotes, wherein my transcription and translation, these two are coupled process and these two are simultaneous also. So we will be having presence of polysomes, right? may attach to mRNA. Of course, they will attach to mRNA, then only they will be able to synthesize protein. Yes, great answer, Monica. They have no role in protein synthesis. They have direct role in protein synthesis. They will attach to mRNA, then only protein synthesis would be happening, all right? So answer is option number D. They have no role in protein synthesis. Clear, Arjun? Yes, Priya. Yes, Tilak. Yes, Rupa. Yes, Ramcharan. Monica. Great. Madhukar, Jogi, Parmesh. Great answer. Next question then. Next one. Who proposed the fluid mosaic model of plasma membrane? Who proposed it? Very common question. And very simple question, I would say. Very simple and very common question. Who proposed fluid mosaic model, according to which we have basically bilayer of lipids, and my proteins are basically embedded inside this bilayer. Protein could be your extrinsic protein, could be your intrinsic proteins, which are basically spanning the two membranes which are there. Yes, Shivoham, Arjun, everybody correct. It is Singer and Nicholson. Singer and Nicholson. Great. Neer, good afternoon, beta. Right? It is Singer and Nicholson. Perfect answers. +4. Moving on to the next question then. Plastids differ from mitochondria on the basis of which of the following feature? Plastids and mitochondria, these two are your double membrane bound organelle. Okay, there are many similarities between these two organelles. Number one is they both divide by binary fission. Number two is they both have double membrane bound organelles. Number three is they both prove endosymbiont theory. Number four is they both have their own ribosome and their own DNA. Okay, they both are semi-autonomous organelles, right? There are so many similarities between these two organelles, but there are some differences also. Tilak has answered yes. Presence of thylakoids. Presence of two layer, of course, they both are bilayer. We have outer membrane and inner membrane. Presence of ribosome, they both have their own ribosome, which is 70S type, okay? We believe that these both organelles used to be prokaryotes once they started living inside the eukaryotes, and then they stayed there forever. Something we call as endosymbiont theory, okay? So they do have their own ribosome, they do have their own DNA also. That is why they are able to make their own protein. But they cannot fulfill their requirement of protein just by making themselves. 50% of the protein they are able to make, but for the rest of the 50% protein requirement, they are dependent upon nucleus. That is why we say that these two are semi, means half, autonomous means independent. So these are semi-autonomous organelles, right, Shivoham? Yes, Arjun. So presence of thylakoid, this is unique to my chloroplast, or I can say plastid in the question, right? But they are not found in the case of mitochondria. So answer is option number C, and presence of DNA is of course there, right, everybody? Yes. Answer is option number C. Shivoham, Arjun, Rupa, Ramcharan. Clear? Next question on your screen, kids. Next question, come on. Next question, come on everybody, solve the question. Yes, cells which are not dividing are likely to be at the cells which are not in dividing phase. Where are they? Great answers. They are into G0 phase. If my cell is into the dividing phase, in that case, first it will go for interphase, then to the M phase. In interphase, we have G1S and G2, okay? So these two stages are for interphase, which means my cell has entered into the process of division. But if it is not, then it will be into G0 stage, all right? Answer is option number C. Perfect, everybody. Next question. Splitting of water is associated with, okay? So we are basically talking about photolysis here. Photolysis, splitting in presence of sunlight. That is associated with what? If I speak about photolysis, then you have came across the funda of non-cyclic photophosphorylation, wherein we have two photosystems. These two photosystems are participating. Yes, PS1 and PS2. Great answers, Rupa, Tilak, Monica, Parmesh, Madhukar, Jogi. Come on, come on. Come on, everybody, keep on try. So basically, inner surface of thylakoid membrane, specifically at PS2. At PS2, right? PS1, not at all. Both of them, not at all. Lumen of thylakoid, no. Inner surface of thylakoid membrane. Correct answer is option number D. Yes, kids? Yes, students? Yes, Priya, Rupa, Grace, Parmesh, Madhukar. Next question, come on, next one. The enzyme that is not found in C3 plants. Okay, so the keyword is the enzyme which is not found in C3 plants. Okay, so we have my C3 plants, we have my C4 plants, right? In case of C4 plant, we have peptase in the mesophyll and rubisco in bundle sheath. In case of C3, we have rubisco in mesophyll and there is no peptase. And there is no peptase. Great, everybody. Answer is option number B, pep carboxylase. See, question is asking you about not found. Rubisco obviously is there in the case of C3 plants. Otherwise, they won't be able to make glucose molecule through Calvin cycle, or we can say C3 cycle. So for C3 cycle, this is acting as an enzyme to fix carbon dioxide. Okay, answer is option number A. But in case of C4 plant, we're gonna see peptase in my outermost mesophyll cells, and rubisco is in bundle sheath cells, right? Yes, yes. Answer is option number 1, which is RUBP, which is pep carboxylase. Sorry, option number 2. Triple R, can you please tell your Insta ID? It is by the name of Bioguru Beena Rao. Okay. Santosh, you wanted to ask something? Santosh? Yes? Okay. Next question then. Come on. The reaction that is responsible for the primary fixation of CO2 is catalyzed by primary fixation. Primary fixation in case of C3 plants, the primary fixation is done by rubisco, clear. But in case of C4 plant, the primary fixation is done by peptase, right? Let's see. So, answer is both RUBP carboxylase, which is rubisco, and pep carboxylase, which is peptase. Clear, everybody? Yes, Monica. Yes, Rupa. Yes, Tilak. Clear, everybody? Is this clear? Come on, students. Okay, do hit the like button also. Do hit the like button and share it also with all your friends. Clear? Perfect. Next question then. Next one. Pyruvic acid, the key product of glycolysis, can have many metabolic fates. Under aerobic condition, it forms what? The end product of glycolysis will decide what? Depending upon the conditions, that whether oxygen is present or not, the fate of pyruvic acid will be decided. Okay, my glycolysis is giving me, from 1 glucose, I will be getting 2 pyruvic acid, right? If there is presence of oxygen, in that case, this pyruvic acid will go for link reaction, then Krebs cycle, and finally to the electron transport chain. But if there is absence of oxygen, in that case, this pyruvic acid will go for fermentation process. Ma'am, in stroma lamella, cyclic phosphorylation, how proton gradient possible? Both sides. Santosh, in case of stroma lamella, just cyclic photophosphorylation will be happening. There is no non-cyclic, okay? If there is just cyclic, which means just ATP will be formed, no NADPH2 will be formed. Is this clear? And the major source of ATP would be non-cyclic, right? Very few ATP we're going to collect from cyclic photophosphorylation, and it will produce just ATP. Clear? Yes. Rupa, Ramcharan, great answers. It is your acetyl coenzyme A plus CO2. Right? Yes, yes. Everybody is correct. Ramcharan, Rupa, Grace, Parmesh, Madhukar, Jogi. Correct answers, everybody. Next question. Mitochondria are called as powerhouse of the ATP. Which of the following observation supports this statement? Why mitochondria are called as powerhouse of ATP? I would say, the simplest question of entire session. Very simple, very straightforward question. Why do we call mitochondria as powerhouse of ATP? Because it will synthesize a lot of ATP. Yes, of course. And why it will synthesize a lot of ATP? Because we have the Krebs cycle happening into matrix of mitochondria. We have electron transport chain happening into inner mitochondrial membrane. All right, we're gonna get lot many NADH2s and FADH2s, which we can convert into ATP. So in total, we can have 36 to 38 ATPs in total. Okay, that is why we call them powerhouse of ATP. Yes, great answers. Arjun, Shivoham, Ramcharan, Rupa, Manikant Reddy, Priya Sharma, Shri Devi Vanala, Grace, Madhukar, Parmesh, everybody is correct. Come on. Next question, next question. A conjoint. Next question, kids. Great answers. Come on, come on. Next question. A conjoint and open vascular bundles. Open, come on, pay attention. Open vascular bundles. Conjoint simply means my xylem and phloem are basically going to stick together. We have one bundle wherein we have both xylem as well as phloem. Open means what? Open means we have presence of cambium. And if cambium is there, which means secondary growth can happen. Conjoint and cambium, where monocot, not at all. Not at all. Monocot, not at all. Dicot roots, or dicot stem. It is dicot stem. All right. So conjoint and open type of bundle you're gonna see in case of dicot stem. In case of roots, we're gonna have radial type of bundles. Radial vascular bundles are there in dicot root. Clear, everybody? Okay, next question on your screen. Match the column type of question. We have column 1 and column 2. In column 1, we have cuticle. Yes, a layer which is, outer side of the epidermis. We have bulliform cells. The cells preferably present into grasses, and curling is happening for these particular cells. Come on, curling is happening for these particular cells. Very important. Then we have stomata. We have epidermis. Come on. Cuticle, guard cell, single layer, waxy layer, empty colorless cell. It is your waxy layer. Bulliform cells, these are my empty colorless cells. Okay? Empty colorless cells, basically reducing the transpiration by exposing or curling down. Basically, if they are curling down, then the exposed surface area would be less. If exposed surface area is less, which means transpiration would be less. If exposed surface area is more, which means transpiration will be more. All right? Yes. So bulliform cells are empty colorless cells, right? Then stomata. In stomata, we do have presence of guard cells. The shape of guard cells vary. In case of dicot, we have bean shape. In case of monocot, we have dumbbell shape, okay? Then we have epidermis, which is your single layer of, which is providing protection to my plant, okay? Let's just match them. A is 3, B is 4, C is 1, and D is my 2. Answer is option number 1. Yes, Ramcharan, Rupa, great answer. Great. Priya, Manikanta Reddy, Shri Devi, Tilak, Monica. Perfect. Next question. Roots developed from part of the plant other than the radicle. If my roots are developed from any part which is not radical, what do we call them? See, if my roots develop from direct elongation of the radicle, I call them primary roots. If my roots develop from any other part except your radicle, I call them adventitious roots. Right, everybody? Yes, Manikanta, Ramcharan, Rupa, Monica, Tilak, Shri Devi, Priya Sharma, Grace, Madhukar, Jogi, the helper, Parmesh is there. Great answer. It has to be option number C, which is adventitious roots. Come on, +4 and move to the next question. This question has been picked up from NCERT. Okay. The giant redwood tree, sequoia, is what? From NCERT, this simple text has been given. In your NCERT, this simple text has been given. the giant redwood tree? Sequoia. Is it an angiosperm? Is it free fern? Is it pteridophyte? Is it gymnosperm? Come on, kids. It is your great answers. Great answers. Great answer. I can see Rupa. I can see Tilak. I can see Priya. I can see Ramcharan. I can see Shri Devi, Manikanta, Monica. It is your gymnosperms. It is your gymnosperms. Correct, everybody. +4. Next question. We have holdfast, stipe, and frond. They constitute the plant body in the case of. Specifically, in some kind of algae, we're going to see these structures, which is holdfast, stipe, and frond. They are the parts of rhodophyceae, parts of phaeophyceae, chlorophyceae, or all of these. Good afternoon, Chini beta. Good afternoon, bache. Yes, yes, come on, come on. Come on, kids. They are the part of phaeophyceae. In phaeophyceae, we're going to have brown algae. Brown algae is there, correct? Yes. Yes, we can have holdfast, stipe, and frond in case of brown algae. Correct, correct, everybody. It is option number C, which is phaeophyceae. Perfecto. +4. Next question. A plant shows thallus level of organization. All right? It shows rhizoids and is haploid. We have given some characteristics of the plant, okay? Number 1 is we have thallus level of organization. Number 2 is we have presence of rhizoids. And number 3 is the plant is haploid. It is haploid, right? Now, it needs water. Very important point. Requirement of water for fertilization. Okay, there is this one plant called as amphibians of plant kingdom because it do need water for sexual reproduction. Okay, now the question is easy. Now the question is easy. It is your bryophytes. Okay, we have given 4 characteristics of this plant. We need to pick up which plant we are speaking about. It is thallus level of organization. We can have 2 options, algae and bryos. It is, they shows rhizoids. Again, we can have 2 options, haploid. Again, we can have 2 options, but they need water to complete their life cycle is very exclusive feature of bryophytes only. That is why answer is option number D, which is bryophytes. Come on, everybody, +4. +4, and next question on your screen. +4, and next question. During light reaction in photosynthesis, the following are formed. In case of light reaction, okay, we have light and we have the dark reaction, right? In case of light reaction, what we can expect? What we can expect? ATP and sugar. Sugar will be formed in light reaction. Think about it. ATP, hydrogen donor, and oxygen. Okay, ATP, hydrogen, and oxygen donor, hydrogen, O2, and sugar. Okay, if I segregate between these 2 reactions, we have number 1 light reaction and number 2 we have dark reaction. Okay, more specifically we call this one as a light dependent reaction, this one as a light independent reaction, right? Now in case of light dependent reaction, what is happening? My plant is going to synthesize the ATP and NADPH to basically energy so that we can actually fix the CO2 into glucose molecule, right? So in case of dark reaction, we are going to fix the CO2 into glucose molecule. So glucose will be fixed into dark reaction, but for that we do need ATP that will be utilized from the products of light reaction. All right, now tell me the answer. Come on, kids. It is ATP, hydrogen donor, and oxygen. Yes. Oxygen donor. Who is oxygen donor, bache? We do have presence of oxygen, not the oxygen donor. All right, so answer is option number C. ATP will be there, hydrogen donor will be there, and oxygen will be provided into light reaction. Come on, next question on your screen. Okay, yes, this is it. Yes, yes, yes. Option number C. Students, you can write me down your marks in the comment section, okay, how much marks you have attained from this particular session. And secondly, if you have any kind of doubt, then please put it into the comment section. We will surely reach out to you and will give you the answer of the question that you do have, all right? And do hit the like button and share it with all your friends and students. We're gonna meet on 5th of May, just after your exam, sharp at 5:30 p.m. All right, just after your exam. All right, students. So we'll see you in the next classes then. Keep your doubts in the comment section. Till then, be happy, be healthy, take care of yourself. See you in the next class. Tomorrow, same time, 3 to 4. Bye, everybody.